And so, as I mentioned earlier, Dr. Annemieke Farenhorst is joining us today, presenting Pesticides in Water and Wastewater, a Prairie Perspective. So Dr. Farenhorst is the Associate VP Research at the University of Manitoba. She has a multidisciplinary research program, including fundamental studies as they apply to better understanding the fate of organic chemicals in water and soil, with practical applications focused on bioped systems to reduce point source pollution by pesticides. She continues to have an interest in environmental science research that is in partnership with First Nations communities. In addition, Dr. Farenhorst collaborates in cross-national between Canada, Sweden, and Germany, and cross-sector, including private, public, and academia, to study women's retention and advancement in engineering and information technology careers. So I will pass the floor over to you, Annemieke.
Thank you so much. Thank you so much, Heather. Thank you also for the length acknowledgement. I'm joining you here today from Treaty 1 territory. Thank you, everyone, for being part of this virtual presentation. And to start out with my presentation, I too want to thank Dr. Nicole Wilson and Claire Herbert for their leadership in this UNE hub for SDG 6. I think they have been doing a wonderful job, and this week they are indeed in New York City to be at the UN 2023 Water Conference. They're representing the University of Manitoba. We have a small delegation going there, and I think it's something that we can all be proud of as the University of Manitoba. So we are indeed the hub for SDG 6, which is clean water and sanitation. And there are a number of goals associated with this particular topic. There are 6 main targets that the UN is trying to achieve. And when I say UN, it's really all of us in the world trying to achieve universal and equitable access to safe running drinking water, as well as adequate sanitation and hygiene. And if we're looking at SDG 6, we think often about clean running drinking water and sanitation, but it's much broader than that, because the targets of the UN and SDG 6 also include reducing water pollution and increasing water efficiency. And I encourage all of you that are here today to check out the website and look at the targets. So you may wonder, why do we still need the SDG 6? Well, over 650 million people on this planet still are forced to practice open defecation. And when I think about that, I particularly think about the hundreds of millions of girls and women that lack privacy when they are menstruating. And they're also, as we know, are at increased risk of sexual violence if they don't have access to a private bathroom. Are there things why this is still needed? Why do we still need to put attention to SDG 6? Is that 1 in every 4 people on this planet, and we have about 7.8 billion people on Earth, 1 in every 4 person on Earth lacks access to safe running drinking water. And in addition to that, there are also billions of people affected in a sense that they do not know the quality of water they are consuming. So we can think about the impact then that this has on people's lives. And there are 2 pictures on your screen. One is very Western focused in terms of its intention, and that is Maslow's hierarchy of needs. And then the other one, actually, I have derived that from Australian and New Zealand water quality guidelines for fresh and marine water quality. And that is an indigenous perspective. So if we are looking whether it's either the indigenous perspective or the Western science of Maslow, We really understand that water is something that we need in order to be healthy. When we think about healthy individuals, healthy culture, healthy countries, self-fulfillment, etc., water is something that is so essential to us that if we do not have healthy water, we are not able to be healthy as individuals and as a society. So when we are thinking about water pollution, often we're thinking about chemicals that we cannot see. We all know that Lake Winnipeg has a concern about nutrient loading. We will see the algae bloom, but we not necessarily see the phosphates or nitrogen that is present in the lakes, nor do we see organic chemicals such as pesticides, UV chemicals, antimicrobials, etc. I think one thing what has changed is the attention to microplastics, for example. And microplastics is something that we see readily in our lakes and rivers. And I think this has to a certain extent helped the society to understand the scope of pollution that exists in rivers and lakes around the world. So I tend to look at pesticides, and we have sampled rivers in Manitoba, and this is an example of studies that we have done in the Red River. So we have sampled all the way from when the river comes in at the US-Canadian border in Emerson, and then we have sampling points along the way up to Lake Winnipeg. Another researcher at the University of Manitoba is Mark Hansen, and he has actually looked for microplastic in this same river, again, starting out as Emerson and then along the way up to Lake Winnipeg. And what Mark found with assistance of some of his students, that microplastics are clearly entering the Red River because of the wastewater treatment system is unable to remove all of them in their wastewater treatment. So this is an example of a P. Muser article that was published by Mark and his student Sarah. And this looks at upstream and downstream microplastic densities in the Red River, and clearly the wastewater treatment plant is a source. One of the things that my students and I were interested in is, what does that mean if we have microplastics in the environment? Can they actually be a carrier of chemical contaminants? There have been substantial studies done on persistent organic pollutants. So these are chemicals that stay around for a long, long time, such as DDT, which is the bottom row in your slide. And there have been a substantial number of studies published that these persistent organic pollutants can sorb very strongly to microplastics. So we have done studies that we looked at absorption of DDT by various microplastics, and these tend to be high, 85% to 94%. When we compare that with current use pesticides, such as 2,4-D, atrazine, and glyphosate, these are herbicides that are widely used in Canada, we did not see the same observation. So current use pesticides tend to not stick to these microplastics, with the exception of glyphosate onto PVC particles. So if we're thinking about understanding whether our microplastics are a carrier of chemicals, we understand that they can be a carrier of chemicals that are very hydrophobic, but much less so to not being a carrier for pesticides that we currently use, which tend to be more hydrophilic. And so here are the a comparison of these four chemicals. You can see DDT has a high log KOW, which means that this is a compound that is hydrophobic, and while the current use chemicals are not. So Canada has about 20% of Earth's total freshwater resources, and this is actually a very small segment of Manitoba. And you can see why we actually calling Manitoba the land of 100,000 lakes. So we have a lot of water in our province. A while back, I was involved in creating an indicator of the risk of water contamination by pesticides. So what we did, we modeled across Canada. We focused on agricultural lands, and you can see in the picture that around 85% or so of the agricultural land in Canada is located in the Prairie provinces. And we started this work quite early on. We worked on it for about a little over a decade, and we started very simple. And towards the end, we including stochastic models in our estimates. And although the math may have seemed quite complicated, as you see on this slide, the main discussion at that time was actually how can we understand and communicate risk to the public. And this is still very difficult, particularly because we do not necessarily know the impact of chemical mixtures on organisms, including humans. And this was also difficult because we did not have appropriate pesticide use data across Canada. So you can model and understand what concentrations might be present in certain water bodies, but it becomes much more difficult to understand where these pesticides are coming from and what their impact are on the ecosystem. We do know that Canada is a large country. We have a significant land mass, and we do apply pesticides in both agricultural and non-agricultural applications. And there are recent estimates that we use between 90 and 130 million kilograms of pesticide active ingredients on an annual basis. And we're actually in the top 10 countries of pesticide use in the world. So most of the pesticides are applied in agriculture. However, there are many different other ways by which we apply chemicals. This includes in our recreational sites, in right-of-ways, for example, and also in our forest industry, and also the generation of hydropower. So estimates are that about 1/3 of the pesticides used on an annual basis in Canada are actually used in in non-agricultural sectors. So we have sampled, as I said, the Red River and other rivers in Manitoba. And one of the things that we do find is pesticide mixtures. So when we are sampling surface waters, over 80% of our water column samples, as well as over 80% of our bottom sediment samples in rivers, do contain pesticide mixtures. Here is an example of what we find in southern Manitoba rivers. This is from 2017, and you can see a range of chemicals are being detected. The numbers refer to the number of detections, and we sampled on a biweekly basis from May to August. And you can see that multiple sites, we detect multiple chemicals and the same chemical over and over again over time. One of the issues when we're thinking about the impacts of mixtures is that we do not even have established Canadian water quality guidelines for individual pesticides. So if we are looking at the 30 or so pesticides that we tend to find in rivers, only 20% of those pesticide active ingredients have an established Canadian water quality guideline for the protection of aquatic life. And if you don't have the guidelines, you do not know whether they are below or above the guidelines, and hence it is difficult to assess any impact. We can look at measures such as lethal concentrations of particular pesticides on organisms. And one that is often used is the fathead minnow that you might know from going fishing. And I want to give you an example about chemicals that we do find up north. For example, 2,4-D, bentazone, and MCPA. We do find that in rivers that are predominantly around forest and wetland complexes. The concentrations that we find are very, very, very low compared to LC50 values for fathead minnow, for example. So when we measure the Manitoba River, we see maximum values for 2,4-D of 591 nanogram per liter. And if we're actually looking at the 96 hours LC50s for a fathead minnow, about 4 these compounds, we're looking at over 100 million nanogram per liter. So mind you, the concentrations that we find of the various chemicals in mixtures tend to be relatively low compared to lethal toxicities that we can find for certain organisms such as fish. We have to realize that over time, our instruments have been improved. So now we are at the PPT level. And for those that are wondering what the part per trillion is, it's actually a single second in 32,000 years. So that painting that you see, that was painted 32,000 years or so ago in a rock formation. If we take 1 second out of all of that time, that's part per trillion. So we can detect very, very, very small amounts now in our waters. If we're thinking about the impact of human activity, including cropland and urban complexes, on the maximum number of pesticide active ingredients detected, it's certainly there. And this is an example of the maximum number of pesticides detected. If we're looking at the Red River that is heavily impacted by human activities, 19 different pesticides in a single sample of water and 9 different pesticides in a single sample of sediment. If we're looking at rivers that are more, I would say, in natural conditions, surrounded by natural condition, that number substantially decreases. So we know for a fact that human activities, of course, have an influence on the number and concentrations of pesticide detected. Interesting enough, we tend to detect a group of the same chemicals no matter where we are located. And these are 2,4-D, MCPA, bentazone, bromoxynil, and chlorpyrrolate. So whether we are high up north sampling rivers or close to the University of Manitoba, we tend to detect all of these chemicals in the waters. Interesting enough, if we're looking at, and these is a screen of over 170 pesticides that we looked at. The good news is that we only detect 30 or so unique active ingredients in the water column and the bottom sediment. So overwhelming number of pesticides that we screen for, we actually never detect in the samples. We still detect legacy compounds, such as transformation products of DDT. But if we're looking at the types of chemicals we detect, it's primarily current use chemicals. Interesting enough, often we sample, if I think about monitoring samples that are done by governments, whether we are in North America or Europe or elsewhere, primarily they're focusing on the water column. However, the water column is not reflective of what we find in bottom sediment. So 15 of the 34 compounds that we detect in the water column, we do not detect in the sediment samples. And 32 of the different pesticides that we detect in bottom sediments, we do not, 17 of those we do not detect in water samples. So if we really want to have a true understanding of what kind of pesticides are present in our water bodies, we really need to sample both sediments and water. These are an example of the chemicals that we frequently find in sediments. So glyphosate, as well as its transformation product, AMPA or AMPA, are chemicals that we detect quite frequently. Here are some of the transformation products of DDT that we still detect frequently, even though we haven't used that since the late 60s, early 70s or so in Canada. And this is an example 2,4-D. We detect it in the water column, and we detect it less frequently in bottom sediments as well. Glyphosate and AMPA, we only detect it in sediments. We never detect it in the water column here in Manitoba. And that's partly because glyphosate, even though it's very water soluble, it sticks very readily to sediment. And so that's one of the reasons why we detect it in the sediments and not the water column. So if we're looking at sustainable development goals, one of the issue currently is that neighboring countries do not necessarily have a good understanding or agreement about transboundary waters. And I think that sometimes or often what happens is that water is moving from one country to another and carries with it pollutants. So this is an example of the Red River. If you look at the bars, these are different pesticide compounds that we tend to detect in these rivers. And their concentrations. And if you see what comes in from the United States, really, we don't see much differences along its pathway all the way to Lake Winnipeg. So some of these chemicals merely come from the United States. Some may degrade along the way when the river is moving to Lake Winnipeg. Other chemicals enter. But overall, what we see is that there's not much difference in the total concentrations of these chemicals along the pathway. And so we have to understand that same with air and same with water, things move from one country to the next, and we don't really have a good collaboration between countries when it comes to reducing pollution of our ecosystems. In contrary to surface waters, we have also monitored groundwater and looked for 170 plus chemicals. Only 3% of the groundwater samples contained mixtures, and that is partly because the soil is a very good buffer. So our groundwater on the prairies is much less contaminated than our surface waters, and only 3% of the groundwater samples do contain mixtures. What's interesting enough, just like we often observe in surface water, MCPA and 2,4-D are two herbicides that we also detect more frequently in groundwater than any other pesticides that we apply in the prairies. And you can see here the total concentrations in some instances exceed 1,000 nanogram. And this is the highest, over 7,000 nanogram. We may ask ourselves if this groundwater is actually used by rural communities as drinking water, as well water. Is that a concern? Well, if we would be living in Europe, they have a regulation that drinking water concentrations, if we're looking at pesticides, cannot exceed 100 nanogram per liters. Or if we have various pesticide mixtures in a sample, it cannot exceed 500 nanogram per liter. If we compare that with our Canadian standards, it's quite a different story in that our maximum acceptable concentrations for some pesticides, individual pesticides, are, for example, 100,000 nanogram per liter for 2,4-D, 280,000 nanogram liter for glyphosate. And this is very, very different than the European system. Well, you may think, is it because the Europeans are more sensitive than the Canadians? Not really. It is because we have different regulatory systems. So the European Union focuses on chemical thresholds. What are the minimums that we typically can detect with our instruments and for chemicals? Well, Canada has a different system, which is really based on toxicity thresholds. So we actually incorporating the animal system in the approach of our regulatory assessments. I can't really tell what's a better system. I can only say it's very, very different. So lastly, I also want to touch upon some of the research that I have been doing more recently, and that looks at biobed systems to turn pesticide rinsate into clean water. So I've worked in Central America in the past, and this is an example. Here you see pesticide handling and empty containers in Honduras. And on the other side, you can see pesticide handling of rinsate. So that's the leftover pesticide solution in your tanks. And that in North America, more often than not, we simply dump that onto an area of the farm over and over again. These are two examples, one of Central America, one of Canada, that actually says these are examples of point source pollution. The disposal of pesticide containers that are inappropriate on the slide here in Honduras, as well as the disposal of pesticide rinsate, leftovers in the tank. And you see that area here, that's the area where rinsate is being disposed of. And as you can see here, nothing is growing anymore. So while we were in Honduras, we worked with communities to develop the recycling of pesticide containers, and that was quite successful. Here in Canada, we do have a good system in place for the recycling of pesticide containers. But I think we still can do much more work in terms of handling rinsate here in Canada. This is a picture of the biobed that we developed on a research farm that is part of the University of Manitoba. And basically, the way that the biobed is working is that it has a cell which contains a mixture of soil, peat, and straw, and it's mixed together, and it acts as a very, very good home, so to speak, for microorganisms. Most pesticides that we know of are degraded by microorganisms. So these are the ones that really help to clean up our environment. And so the pesticide rinsate is collected in a storage tank, and then it's drip irrigated to these biobed cells. Here you see the drip irrigation, and then the microorganisms in that biobed do the work. They start degrading the pesticides. Some pesticides will also retained by this organic matter rich substrate. And as a consequence, the effluent will be much cleaner. This is a picture that summarizes that. So we have the rinsate storage. It moves into the first cell, then it moves into a second cell, and where the treated rinsate is then collected. This is the rinsate. You can see it's quite cloudy, moving into the first cell. This is midway, and you can see here that what comes out of the second cell is really quite clear. And if we would look at chemical concentrations, this is an example of the various chemicals that were applied on the farm. This is sampling during 2021. You can see here a range of chemicals were applied. So this goes into the biobed, the effluent cell number 1. still has a number of chemicals, but at a much, much lower concentration. And there are also fewer chemicals compared to what went into the influent. And then if we're looking at the effluent cell number 2, only 2 chemicals left, again, at smaller concentrations. So this shows that biobeds are effective for a lot of chemicals, but not all. So we also have collaborated with Agriculture and Agri-Food Canada on biobed research. Here shows the biobeds that we have looked at, single cell biobeds, so only 1 cell, and dual cell biobeds in Saskatchewan, just like the dual cell biobed in Manitoba. And so data were collected for a number of years, and this looks at various chemicals and the percent reduction due to the biobed. So you can see here that for many of the chemicals, there's a close to 100% reduction in their chemical concentrations. And you can also see that both single cell and dual cell biobed seem to be effective under prairie conditions. We can also look at what does that mean from a toxicological point of view. And this is a picture that shows the pesticide toxicity index. And basically, you look at pesticide mixtures in a sample, you compare it to benchmarks in a acute toxicity database. You're looking at sensitive species, and you use an additive toxicity model to come up basically with a pesticide toxicity index. This shows a dual biobed cell influent, what goes in, and this is what ultimately comes out after the second biocell. The pesticide toxicity index, this is 1200, the y-axis, here it's 120, so there's a 10-fold reduction there. Here you can see that whether we look at fish, toxicity to fish, invertebrates, or plants, we can see that the influent, the pesticide mixtures have a high level of toxicity. However, once the rinsate has moved through the biobed, a much, much lower pesticide toxicity index remain for regardless of what organism you look at. So I think biobeds can have a benefit to our ecosystem under prairie conditions. So this is another picture. Now I show also the single cell. So the single cell here, dual cell here, and as you can see for both single and dual cell biobeds, pesticide toxicity index drastically reduces once the pesticide mixture rinsate moves through the biobed. So with that, I thank you very, very much for listening to the presentation. Some of the data that I have presented here today is thanks to the people that you see here on the screen. This is a picture taken pre-COVID. Nobody has masked, but I want to thank all the graduate students and the summer students and research associates for their hard work in collecting all these data. We have established during COVID a whole new lab. We call that the MASS lab. It's located in the Smart Park, and so we currently analyze for pesticides. As I mentioned already, we look at UV chemicals, antimicrobials, some other compounds. So if you are in that line of work and have an interest in those analysis and don't have access to equipment yourself, we're always open for discussions. And lastly, we thank our funders. The work that I showed you today was primarily funded by NSERC, the Canadian Agricultural Partnership, and also Bell MTS. And I want to also thank my students for being such a good sport during COVID. I know it wasn't easy, but definitely they continue to have high interest and energy in research. So thank you for that as well. And with that, I happy to address any questions that you might have.